Hi, everyone. Good afternoon. Thank you for sticking around till the end. So I'm Ethan Wolfe. I work in the J. Ravi lab at the University of Colorado Anschutz Medical Campus. I'm entering my senior year of undergraduate at Michigan State University, but I've been working remotely, and it's really a pleasure to be able to give this talk here today. So my research that I'll be discussing is leveraging machine learning models to predict antimicrobial resistance in the ESKAPE pathogens. And I think I'll go ahead and get started. So antimicrobial resistance has really become a global public health crisis. And this has stemmed from the continued misuse and overuse of antibiotics in healthcare settings and agriculture. And this misuse and overuse has allowed these bacterial pathogens to evolve antimicrobial resistance mechanisms. According to the US CDC, there were 1.27 million deaths directly attributable to AMR in 2019 alone. And so this is sort of, that kind of tells you that we're in a crisis here. And our main question is, how do we understand and address this crisis? Well, we need to start with the million-dollar question in biology, which is how do we map the genomic features of ESKAPE to an antimicrobial resistance phenotype? So in this research, we're interested in doing just that, associating gene presence absence in ESKAPE to an AMR phenotype and identifying the top AMR associated features. And to do this, we constructed pangenomes per species and trained machine learning models on these. So then comes the question, how do we construct a pangenome that's suitable for machine learning? We first start with the collection of all genomes of a single ESKAPE member from NCBI. And these are annotated with PGAP, the prokaryotic genome annotation pipeline. We then create homologous gene clusters with Panaru and create a binary genome by gene presence absence matrix, which is our numerical representation of the pangenome. And we have AMR phenotype labels per genome, denoting resistant or susceptibility to a given drug. And that's from BVBRC, the Bacterial and Viral Bioinformatics Resource Center. Okay, now getting back to this question of how do we map the ESKAPE genotype to the AMR phenotype, we work in our studio using the Tidy Models package for machine learning, and we start by splitting our pangenome dataset into training and testing. We perform hyperparameter tuning on the training set using tenfold cross-validation, and based on our optimal hyperparameters, we fit a final model to this training set. And we're able to obtain a ranked list of genes that our model has associated with antimicrobial resistance based on the presence-absence pattern. And then we apply this to the testing data set where we hide the actual phenotype label from the model, and we ask it to make predictions on the test data set based on the gene presence-absence pattern. So we can then evaluate the model predictions with various performance metrics by comparing the actual phenotype to the predicted phenotype. Let's look at a case study of how our logistic regression model performed in Enterococcus faecium. So for vancomycin, we had 237 Enterococcus faecium genomes with AMR phenotype data. And you can see these are all lab derived, and the class balance here is pretty alarming, if I do say so myself. And so for the model performance, we looked across, we have a few metrics, but here we're looking at AUPRC and balance accuracy. Interestingly, the models perform astonishingly well with AUPRC, but not quite so well with balance accuracy, which is curious. And, this could prompt us to think we need to look, seeing this in other cases too, we need to look beyond just gene presence absence and look at other biological scales, molecular resolution, to improve these models. So we did this across multiple drugs, multiple drug classes. And within Enterococcus. And we can see that irrespective of the class imbalances, in some cases, the models perform pretty well at their classification task. And so for Enterococcus faecium, the top vancomycin resistance genes predicted by logistic regression were none other than Van X, Van Y, Van Z, Van H, and Van R. No surprise there. So the models are doing well in predicting known AMR genes. So now we were curious what would happen if we trained a model and got a ranked list of top AMR associated genes. What would happen if we removed these genes from the pangenome and retrained the model on the smaller pangenome? And then we kept doing this recursively until we had a smaller and smaller pangenome. How would our models perform? So with the glycopeptides, teicoplanin, and vancomycin, this did exactly what it, as expected. As we dropped the first top 200 genes, immediately the model performance fell off, and it was essentially a random classifier. It was doing very poorly, and this was to be expected. But for the other antibiotics on there, gentamicin, linezolid, streptomycin, this wasn't the case. We had removed nearly half of the genome before the model performance started to drop significantly. And this was really an interesting find from our research. We didn't expect to see this. So a few conclusions that I want to draw here. So the supervised machine learning reliably was able to predict AMR from gene presence absence in some of the most notoriously drug-resistant pathogens, ESKAPE. And going back to the recursive top feature elimination, it was really interesting to see how the models continue to perform well, irrespective of having lost a bunch of genes. And this could suggest that organisms need to modify more than just a few genes to adapt to selective pressures in their environment. So in the future, this research could greatly benefit from dimensionality reduction given the high gene to genome ratio. And we're also very interested in cross-species analysis. So, how do we make these homologous gene clusters comparable across different species within ESKAPE? And as I mentioned earlier, we're also very interested in going beyond genes, gene presence absence. So, in the literature, you'll see SNPs and KMERs as common examples. We're also interested in protein domains, which Charmy in our lab has been working on recently, biological pathways, genomic context. All of these molecular scales are really important in thinking about this problem, and thinking about ways to integrate these is going to be key to better understanding antimicrobial resistance and the molecular mechanisms behind it. So with that, I'd like to thank all of the wonderful people on our AMR team, and all of the past contributors. There's been a lot of people who have been working on this project. So thank you so much to all of you. And thank you to all of you for your attention. I greatly appreciate it, as well as the ISMB Travel Fellowship that I received through EvolCompGen. So that QR code will take you to the lab website. Thank you so much. I'm happy to answer any questions.
We have plenty of time for questions. Well, if nobody has a question, I have a question.
Okay.
Yeah, so I may have missed this, but when you were talking about, so you have a number of features that you look at. Do you look at, is one of the important features for predictions, looking at things like, is it present on a plasmid, or are there other features that you find particularly helpful?
Yeah. So as you mentioned, presence on a plasmid is kind of huge. We didn't just look at Enterococcus faecium, we went across ESKAPE. So we saw a lot of examples where like 9 out of 10 of the top gene hits were plasmid associated. So that means there's really a lot of horizontal gene transfer going on here. And I think one of the previous talks today alluded to the fact that 84% of the antimicrobial resistant genes were associated with or located on genomic islands. So that's Yeah, definitely. And then, as I mentioned, we'll need to look at those other molecular scales to get a more full picture of this.
Yeah, thank you. Great talk.
Thanks for your question.
So my question is somewhat related to that, which is when you started eliminating these top cases, and you showed many of these curves where the model was still performing quite well. Do you have any insights into what those genes were after you eliminated the very top cases? Did you look at those, and what were those genes that were still contributing to the model?
Yeah, that's a really excellent question. That's definitely something we're very curious about. We're very curious to see, how these are, like, anything about these that we can learn would be really interesting, I think. But because we have the transfer of genomic islands being so involved with antimicrobial resistance, perhaps these islands can be quite big, and perhaps there could be something to be said there, where we're just getting genes that are so moving around with an island, and we're just looking at presence absence, and that's why there's so many that seem to be quote unquote associated. But when we're seeing half of the genome, being like seeming to have an effect, then that's really puzzling. So,
yeah, thanks. I was just wondering about the pangenome matrix, and is that just the binary presence-absence matrix of genes, or is there any other information in the matrix as well?
Yeah, so it's just a binary presence absence matrix based on the homologous gene clusters across within a given species. We haven't integrated other levels, and you can think about maybe doing a similar approach with k-mers or with protein domains for that.
Okay, thanks.
You're welcome.